Bio 100. Okay, welcome back. So hopefully you've had a chance to think about what type of research question you're interested in, as well as the types of variables you might use to answer that question within the framework of a testable hypothesis. So to recap, the general stages of scientific problem solving or coming up with a research question and addressing it is the observation or looking at some kind of natural phenomena or phenomenon that's interesting to you or sparks some curiosity for you, and coming up with some kind of explanation for what you're observing, and then devising or coming up or creating with some sort of test or experiment that will help you accept or reject your explanation, in other words, your hypothesis. And then you actually have to go through and do it. You have to conduct the test and statistically test, in most cases, your hypothesis or explanation. If you fail to reject the null, or in other words, accept your best explanation, then you go back to the drawing board. You come up with another explanation and you start the process over again. Maybe there was a variable you didn't consider that you should the second time around. So as an example of this process, I'm going to use an example of Mr. Smith, who was going to France for the summer and kind of wanted to play around with using the French language instead of English with his dog, who, just for the sake of simplicity, is named Dog. And so rather than saying walk or walkies, he decided to use the French word Allons. And so he noticed very quickly that Dog understood when he said Allons and came running because he was excited to go on a walk. And so what's the observation here? And what's a testable explanation? And is it the only explanation? So the observation here is that the dog responds to Allons. When Mr. Smith calls Allons, the dog comes running, just like he would if he said walkies. And so Mr. Smith's explanation for this, or hypothesis, is that the dog understands Allons as meaning walkies. In other words, he understands that French word. The real question now is that the only explanation? And it's not. The dog could be responding to other situations or cues that are carrying along with Mr. Smith saying walkies or Allons. It might always be after dinner. It might always involve walking to the door or putting a coat on, or just the act of calling in a certain tone of voice. And so the actual word Allons may actually only be a small part of the cue that triggers the dog to think that it's going to go on a walk. And so it's important to consider all these alternative explanations if you wanted to, for example, test the dog's understanding of the French language. So this is where experimental design comes in. Can we devise some sort of experimental test that will allow us to discriminate between the different possibilities, or at least eliminate those other possibilities as we test each potential explanation? For example, one way that might correct for one possible explanation is, rather than always calling Allons or walkies to the dog after dinner, maybe you randomize it and call Allons before breakfast, or at noon, or 1 or 3 p.m., and mix it up and see if the reaction is the same. That would eliminate the explanation of it being a cue based on timing of the call. Or perhaps a control for whether walking to the door prompts the response in the dog. Maybe have a part of your experimental design where you walk to the door with a coat on without saying anything and see if the dog still responds. Or do everything as you normally did, but instead of saying Allons or walkies, say something totally unrelated like rhubarb or potato and see if the dog responds. So by doing this, you can kind of control for some of the other explanations. And so if you were to kind of do these 3 or 4 different ways of ruling out alternative explanations, if you still saw that the dog responded to Allons, it might actually give you support for your explanation that the dog is understanding that that French word is synonymous with walk. What it does not tell you is that how far you can extrapolate that explanation. So we very thoroughly tested that the dog named Dog understands Allons means walk, but can we extend that to all dogs? Do all dogs understand that that word means walking? Or is it just specific to what you perform the test on? And this is kind of where you as a scientist have to make judgments about how far can you extrapolate the findings from any given experiment. So I gave you an example of some potential experiments to rule out alternative explanations, but let's talk a little more about how you design experiments. And so, as we used in that first example, it's really important to have some level of discrimination in your experiments. In other words, discriminating between alternative explanations that you're not hypothesizing explain the phenomena you're interested in. And so the design of an experiment to test a hypothesis really kind of takes a lot of, in many cases, creativity. There's not a one-size-fits-all answer. Sometimes what makes a great scientist great is their ability to come up with clever experimental designs to tease apart alternative explanations that people haven't been able to do before. Along with this kind of creativity, typically is a suspicious nature, not taking anything at face value, kind of questioning everything. And while in modern biology, most experiments use very sophisticated equipment, sometimes the most clever experimental designs can be quite simple. And so regardless of what types of equipment or you are or may not be using, there are some features that are common to all good experiments and that you typically won't find in bad experiments. And so discrimination being one of those, you want your experiments capable of discriminating between different hypotheses, because if you don't do that, then at the end of the day, you have results that don't really tell you one thing or the other. They don't get you any closer to understanding the underlying explanation for some observed phenomena. The next thing most good experiments tend to have in common is that they tend to have some measure of replication and are built in a way that they can be extrapolated a little more generally. So living organisms, which is the subject of most biological research, are variable. I am different than all of you, even though we're all humans. I'm different from my sibling or from my parents. And so there's variability. And so if you wanted to measure the circumference of a human head, you wouldn't just measure mine. You would measure many, many people to get an idea of the variance. And so typically experiments are repeated as far as their measurements go many times. And because of the variability of biology, we need to be cautious about how we generalize our results from individuals to members of a group or population that we're studying. So anything we found on the example of dog would not be reasonable to extrapolate to all dogs or all mammals or all vertebrates. Now, if we took 500 dogs and we taught them to respond to the word Allons, and we saw that they all in general tended to be able to understand that it was a synonym for walk, then we might be able to then extrapolate that dogs are capable of learning French. And so where your replication is, whether it's replication of a single individual, saying Allons to that dog over and over again many weeks and seeing how it responds versus saying walks or saying nothing or rhubarb or potato, are a bunch of replicates of the same individual and therefore cannot be extrapolated reasonably to all individuals of that species. But if your replicates were of many individuals within a species, then you might be able to extrapolate there. And so that's where replication really comes in. The other thing most experiments tend to have in common are controls. Many experiments that are comparative need to have a control group. Descriptive ones, maybe not so much. But if you're interested in comparative work, you need to try and you need to check the possibility that other factors in the overall experiment are not influencing your variable of interest. So, as an example, growth hormones are secreted in response to a whole variety of things, one of which is the amino acid arginine. And so there was a study a long time ago where volunteers were injected with arginine. And being good researchers, the researchers said, hey, we need a control also. And so they injected the volunteers with a saline solution as well as kind of their control group. What was interesting, despite them thinking about alternative explanations, they saw that growth hormone was secreted both with the arginine injection and with the saline injection. And so they thought, okay, it doesn't really matter. It's the act of injecting something that prompts the growth hormone response. But then they added another control group where they just took a syringe and waved it in front of the volunteers' faces and they saw a growth hormone secretion again. And because they kind of kept doing all these different controls to remove things that may be impacting the measured variable growth hormone secretion, they found that actually growth hormone is just secreted in response to stress. And so control groups are very important. They kind of help you design an experiment that actually tells you something useful or doesn't tell you something useful. Something else that you need to consider are biases, particularly where humans are involved. Humans are inherently biased, and so this is why you sometimes hear of studies as being single blind or double blind. And the real difference here is in a single blind study, the people receiving the treatment don't know whether they're receiving the real treatment or the control. So many times these kind of comparative studies for drugs are one group is given the actual drug and one is given like a salt pill, just so they get the act of taking a pill to rule out kind of the impact of the mind on making people think they're feeling better. And so these people don't actually know if they're taking the treatment or not, so they are unbiased in their response to it. And so that would be one group is blind. The treatment people are blind. You also will hear about in medical studies double-blind studies, because you could imagine a situation, a single-blind study, where this doctor here really wants this medicine to work. And so if he's measuring something like energy level or something like that, something very subjective, without meaning to be biased, this doctor might just view or score these treatment groups that took the actual drug as having more energy because that's what his or her expectation is. And so in cases where there's a lot of subjectivity in evaluating the scores of response to a treatment, there's typically a double blind that's applied. So not only are the patients blinded to it, but also the doctor as well does not know who got the control or who got the treatment pills. And so this kind of is one way of kind of eliminating some of the biases. You won't typically see the term double blind in studies where the patients are not humans. So if you're looking at plants or rats or mice, typically it will just be referred to as blinded if the researcher doesn't know, because it's pretty much assumed that the subject, the plant, doesn't know whether it's getting the placebo or the control. So typically if you just see blinded and it's not with humans, you can assume that it's the researcher that doesn't know, which is the opposite of kind of a typical single blind setup. Okay, so let's kind of work through an example that you're going to kind of do eventually as well. So let's say that I mentioned this in the last video, I had made some observations and I was eating at Riverfront Cafe and I saw that there were lots of wasps there. And I noticed that there seemed to be a number of people eating salami for lunch, and I thought I saw more wasps bothering the people with salami. And so I wanted to test whether wasps were more attracted to lunches that contained salami than ones that didn't contain salami. And so I need to really define the objectives and write down precisely what I want to test in the experiment. And so if I want to test whether wasps are more attracted to salami than non-salami meals, I need to devise a strategy, design an experiment. I need to think about the size and the structure of the experiment. How many treatments, how many replicates, what variables am I going to measure, and what will be the appropriate statistics. Maybe I'm interested in 2 categorical independent groups with some continuous dependent variable. Maybe I'm going to measure the number of wasps that visit lunches with salami and without salami. And each of my replicates is going to be a different person eating lunch. And so, I might get 5, 10, 3, 4, and maybe here I get 1, 2, 3, 2. Now I've got it set up for, this would be something that would be appropriate for a t-test. What if I decided to go a step further And I was like, I might also be interested in whether turkey salami had an impact. And so now I've got three groups. And so now I'm looking at something like an ANOVA. And so it's important to think about all the different things you're going to measure before you do your study, because this informs me what kind of statistical test I'll be doing. Will I be doing a t-test? Will I be doing an ANOVA? Maybe I'm interested in measuring two continuous things. Maybe I want to look at grams of salami versus duration of wasp visit. And so, here I might actually be doing a linear regression. All three of these are valid ways to test whether salami content of a lunch impacts whether wasps want to visit, but they're all tested in different ways, and it depends on what and who I measure that facilitates which statistical test I'll use. So once I've kind of settled in on the groups I want to analyze, the variables I want to measure, this is where you actually need to really get down to the nitty gritty and come up with the operational details. So what order will things be done? Will I assess everyone's lunch first and then count the wasps? Will that impart bias? If I knew a lunch had salami in it, would I watch it more closely and notice more wasps? Or should I kind of count all the wasps on all the people around me and then go look at what they had for lunch once I'm done counting? How long should I count? Should I observe people eating for the duration of their whole meal, or just for 10 minutes? Should it be in the middle of their meal, at the start of the meal, 10 minutes after they start? These are all kind of things that may influence whether the results of your study are going to provide a good explanation or not. It might be that wasps do respond to salami, but it takes them 20 minutes to smell it. So if you measure the first 10 minutes, you might miss that it's actually influenced by it. How will you randomize the treatments? Will you just walk through the tables outside Riverfront and just kind of randomly pick 5? How will you pick them? Or will it just be the 5 that happen to be next to you? What if you smell weird and you attract the wasps? So no matter where you sit, the wasps always come. So these are the important things to think about. Should it be done during just one season or in multiple seasons? Does temperature of the day matter? So these are all kind of things that you have to think about. If it's really cold out, maybe the volatiles in the salami don't get into the air, and you need it to be hot out where that salami gets kind of warm and melty a little bit. Okay, and so that's like a lot of things to think about. There's so many millions of things that could influence your study. No experiment is perfect, though. There's always going to be gaps or downsides or biases or pitfalls, no matter what anyone does. What's important to consider, though, is not that your experiment is perfect, but it's that you've identified where these potential holes and pitfalls are, and that you outline what those may be so that when your experiment's done, you know what's fair to extrapolate, what's fair to interpret, so you don't go too far. If you found salami near Riverfront triggered a significant increase in wasp activity through one of those experiments we talked about, could you extrapolate it to all wasps? Could you do it everywhere? What about the same species? Probably not. Could you, within reason, try to extrapolate a bit using just your existing knowledge? Well, you might have done some research and shown that the wasps at Riverfront have a really closely related species that have typically a shared diet with the riverfront wasps. So it might be fair to extrapolate that if the riverfront wasps liked salami, that this other closely related species might. And so that might be a fair extrapolation based on kind of your understanding of the science and the organisms.